Welcome to today's European Seed Innovation webinar series. My name is Marcel Bruins and I serve as Editorial Director for European Seed. And today I'm happy to be your host. Today's theme is KASP technology accelerates rice improvement in South Asia. Before we begin, I'd like to take a minute to thank LGC Biosearch Technologies for partnering with us on this Innovation Series webinar. Presenting the webinar today is Catherine Steele. She is a senior lecturer in sustainable crop production at Bangor University in Wales. She studies the genetics and genomics of rice, potato, tomato, and barley to improve sustainable production. She has published over 30 papers and articles and has over 20 years of experience in managing international projects with diverse partners. She has led DFID-funded projects targeting agricultural development in Asia and projects for DEFRA and the Food Standard Agency on basmati rice authentication. Currently, she is the academic lead on an Innovate UK project developing new markers for genomic selection in rice. During the presentation, you'll likely have some questions for our speaker. Please type these into the chat box at any time during the webinar, and we'll address them during the Q&A session after the presentation. Today's webinar is being recorded and will be made available at european-seed.com following this webinar. Catherine, the floor is yours.
Thank you, Marcel, for the introduction. Hello, everybody. I'm Catherine Steele. I'm coming from Bangor, North Wales, and I apologise if you hear any seagulls, but hopefully they won't interrupt us. So I'm going to talk about two projects that were funded by the UK Department for International Development through the Agritech Catalyst Programme of Innovate UK. They're multi-partner international projects with LGC Biosearch as the lead industrial partner and obviously Bangor as the lead academic partner. And the aim was to develop and use KASP markers to accelerate rice improvement, focusing in this project in Nepal initially, and then Pakistan, and also Skewast Kashmir in India. So rice is grown in many countries worldwide across diverse ecosystems and diverse production systems. It's even cultivated by subsistence farmers, and some of those farmers have not been able to adopt modern varieties, and some of them still prefer local varieties because they are better under adverse conditions such as drought. It's important for production to keep up with the demand. In this graph on the left, you can see the utilisation of rice in red going up, and then the production is the bars underneath. And production has kept up with demand over the past few years, but it's going to become more challenging. And as you can see on the right, production is different in different countries, and also actual production in 2017 was affected by adverse conditions such as floods and droughts in countries such as Bangladesh, Sri Lanka, Vietnam, and Nepal. Nepal suffered very much from flooding. And so because of these adverse conditions and also the need to improve productivity and at the same time reduce fertilizer and pesticides inputs, it's really important that we as rice breeders can improve the varieties so that they will withstand these adverse conditions and still be able to maintain the yields that we need in order to meet that demand of the increasing population. The most common targets for selection by rice breeders in the countries with which I've worked are resistance to rice blast, and this is a fungal pathogen of which there are over 100 race-specific R loci known, but of those there's about 20 that have been cloned as R genes. This disease gives 100% yield loss in certain sites. And as you can see from the graph at the top, showing the severity of blast across different states of India, it can vary in different places and in different seasons, up to 100% yield loss. The next most common trait for breeders to select is resistance to bacterial leaf blight, known as BLB, or just bacteria blight. This is obviously a bacterium, and as a consequence, it's very difficult to control with chemicals. So breeding and genetic resistance is absolutely essential for this pest and this disease. There are, again, many race-specific R genes, resistance loci, known. About 6 have been cloned for BLB resistance. There are currently microsatellite markers and specific PCR markers for a number of these R genes, and they're used by breeders already. And in fact, rice varieties have been released in all of the 3 countries on this slide that have been selected using molecular markers for these disease resistances. The third most important target in these countries is aroma. Aroma, or fragrance, in rice adds value, and it widens the export market for many countries. So it can produce higher prices, and it offers farmers added security if they can get more money for their product. The most common fragrance allele in Asian rice is known as BADH2.1. This is found in a gene on the fragrance gene on chromosome 8. So it's a major component of the fragrance in rice. And many PCR markers, well, specific PCR markers are widely available for selection of this allele currently and are being used. But this allele is not the only locus for it's not the only allele in the fragrance gene, and it's not the only locus affecting fragrance in rice. There are other variations within that gene on chromosome 8, and there are many minor QTLs. There are at least 2 minor QTLs on other chromosomes that control aroma. So these are all relevant markers that we've developed as KASP that can be used in the breeding program. And you can see on the right a cross that's being conducted at the moment by our commercial partner in Nepal, Animal Brew Private Limited, APL. APL have been working on a cross between Sonola Saganda, which is fragrant, and which is one of the rice varieties that we used in the project for generating a whole genome sequence. That's been crossed with another variety that hasn't been sequenced, but using KASP, we've found markers that are polymorphic and that have been used for selecting aroma in that cross successfully. There's a huge amount of genetic variation in rice, and this is apparent in the polymorphic diversity of physiological traits and morphology. As well as phenotyping, you can find diversity by comparing rice genomes. So rice genome sequencing has become much more common in recent years, and we now know that there are nearly 40,000 protein coding sequences in rice, code for genes that affect traits. There are also about 35% of that rice genome which is made up of repeat sequences, and often the repeat sequences are not in the genes. And one important type of repeat sequence that you may be familiar with are simple sequence repeats, also known as microsatellites, or SSRs. And there are about 48,000 SSRs across the rice genome. So when you actually compare between any individual pair of rice varieties, you can actually look, if you've got the full genome, you can actually look and find out how many are polymorphic. So approximately 4,500 SSRs will be polymorphic between 2 rice varieties. And then at the sequence level, there are two types of variation that we're interested in. There are indels, which are a short segment of sequence that is either inserted or deleted when you compare two different sequences, and there's about 40,000 of those, and SNPs, which are single nucleotide polymorphism. And of those, between any two rice varieties, there's over 300,000 SNPs. So this shows us that indels and SNPs are much more common in the genome than microsatellite SSRs. So when we come on to compare microsatellites with KASP, so KASP technology is technology that can identify and genotype SNPs and indels, whereas microsatellites are technology that identify variation in the simple sequence repeats. When we started this project in 2014, there were about 18,000 microsatellites available on the Grameen website, and in comparison on the LGC website there were just over 2,000 KASP available. Those KASP had been developed through projects run by the Generation Challenge programme, and they were polymorphic between Oryza sativa and Oryza glaberrima, and were useful in rice breeding work that was developed in that project. And their sequences were used in arrays originally, and they were converted from sequences used in arrays into KASP, and the Japonica reference genome had been used in their development. So the paper at the top right there by Semigan et al., they looked at cost efficiency compared with other rice systems, and they considered KASP to be cost effective compared with, for example, Golden Gate, which is another similar type of technology. But comparing them with microsatellites on the left here, microsatellites are very labour intensive and have high running costs. If you run them in your own lab, you have to use many steps that are repetitive, and you have to use gels. You can use chemicals such as acidium bromide for staining the gels, and toxic chemicals if you're running high resolution gels. They can be automated, but again, that takes a lot of time and effort to get them set up. And the other thing is, as I mentioned on the previous slide, there are fewer possible microsatellite regions in the genome that can be used as markers in comparison with indels and SNPs. So going down on the right, there are numerous SNPs and indels, and they've already been shown to have a high conversion rate into KASP markers. They're very safe to use, and you can use them KASP in your own lab. There's a unique chemistry for running them, and you can use plate fluorescence to detect the results. So all you need is a PCR machine and a plate fluorescence reader, or a real-time PCR machine. But if you don't have your own laboratory, and this is an example that we've worked with a company, APL in Nepal, who do not have a lab, and they have been using the extremely high throughput service run by LGC Biosearch, and this means that you can get your results back just on an Excel spreadsheet. So all you need to do is send your leaf samples to LGC Biosearch, and they do the extraction and the assays and provide you with the results. So the options for running KASP are wide, and they have the benefit as well of being flexible in numbers compared to array sequences, where you're basically stuck with the markers on the array, and many may not be polymorphic in your particular cross of interest. This slide shows the components of the assay. So there are three components. You have the primers. So there are three primers, two for the target that can either be the two bases for that particular SNP, So we've got C and A shown on this slide, and a reverse primer that is common. In the master mix, you have these oligonucleotides. So there are two separate oligonucleotides that have different fluorescent dyes on them. In this case, they're labeled FAM and HEX, and those fluoresce depending on the binding of the two forward primers that have got the two different possible alleles in the sequence. So the amount of fluorescence in the PCR product at the end of the cycle depends on the number of successful binding between either of those two primers. So if only one has bound, if it's homozygous, you will get full fluorescence of one dye. If it's homozygous for the other one, you'll get full fluorescence of that dye. And if it's heterozygous, you get equal fluorescence. And obviously, you need to put your DNA sample template into the reaction. So with KASP, there's various options. If you know what the sequence is, you can submit the sequence to LGC and they can design the primers. If you don't know those sequences, then you can choose them from the database of available KASP. So what we needed to do was work through these series of steps to design and generate new KASP that would work in rice to increase the number of KASP available. So basically, you need to have the sequences either side of the target SNP or indel, which are shown inside the square brackets on this diagram. So the top sequence example shows a SNP where you've got an S. Now the S is the ambiguous DNA code for C or G. So that indicates what the SNP is. But then, for example, you've got a number of N bases which could be variable. So that would need to have, and those are in the flanking sequence. So those would also need to have an ambiguous base put in for the KASP design to work. The second example is for an indel, which you either have the sequence or no sequence, which is represented by the dash. And again, you can see ambiguous bases, S and Y in this case, in that sequence. So once LGC get those designs, they run them through their software called Kraken, and that tells them whether that primer should work in silico, and then they validate it by running it in the actual PCR with, in this case, a rice template DNA. One of the other things that we did in our project was to work out a way of identifying KASP that were located near to SSRs, because SSRs, as I said, are repeat sequences, and they're not the same type of DNA sequence as the SNPs and indels that KASP use. So you can't convert directly, but you can convert in terms of identifying a potential KASP in the flanking sequence of the SSR primers. So in this example, the SSR primers are shown in red, and the potential KASP target are shown with the red stars. So in this protocol, if you know which sequence, which primers you are interested in finding or converting into KASP, then you LGC can sequence either side of that using long-range PCR and next-generation sequencing, and then design assays from that. And we showed in our project that this is highly efficient and works for targeting certain regions. So our project, it started in 2014 as an early stage feasibility project, 2 years worth of funding. And the idea was to develop KASP that were relevant in rice crosses being done by our Nepalese partners. And they were carrying on with the field screening and doing the breeding. So we took information from previous projects that we'd worked on that had led to novel rice varieties that were released in Nepal. We had field data from the Nepalese Agricultural Research Council. We had resistance genes and sequences that are available in the literature, and the KASP technology from LGC. The aims of that project were to assess the potential of KASP for rice breeding in a real life situation, working with breeders, both public sector in NARC and private sector in APL. And we wanted to use whole genome sequencing to start off with to identify variations in breeding lines that were chosen by those breeders and develop KASP that were useful for breeding for the traits of interest to those breeders, and to actually demonstrate the use of KASP in Nepal in a public sector breeding program. At the start of the project, as I said, around about 2,000 KASP publicly available from the Generation Challenge program. And these primers, these markers, when you actually looked at them in the 9 lines that were chosen by breeders, the graph on the left shows the 12 chromosomes of rice, and the coloured squares show where those markers mapped on the genome against the indica reference. The indica reference used here was 9311. And you can see that the highest number of markers in any one of those squares is up to 6, or just over 6 markers. And there are many squares that are white with no markers. In total, there were 1,491 of the pre-existing KASP that aligned to the indica genome with 100% hit rate. And in any pairwise cross between those two, between any pair of those 2 9 lines, there were between 284 and 414 assays. So this meant that there were large chunks of the genome where we didn't have KASP existing to start with. So we used those 9 sequences to look to see how many potential KASP there were. And in this slide, you can see what we found when we looked for KASP, potential KASP, so SNPs and Indels in those. And in that, you can now see that the scale has changed, and the smallest category is between 1 and 10, and there are no white spaces. So we were able to detect between these 9 sequences, these 9 whole genome sequences, that for any pairwise combination between those individuals, we would be able to find KASP within any region of the genome. The 9 parents that were used are shown on this slide in a diagram showing the donor parents along the top row, and they all came from the breeding program of the Rice International Research Institute, International Rice Research Institute, IRRI. And then the recurrent parents on the left side are varieties that were released in Nepal, and they carried no known resistance genes to blast or bacterial leaf blight. And so the idea was to make crosses between those and back crossing using marker-assisted recurrent selection to identify the target, the lines carrying the target alleles for selection. When we sequenced those 9 rice varieties, which are shown on the left here, we used Illumina paired end sequencing, and we mapped the reads against the 9311 reference genome of indica. So this, to my knowledge, was the first really detailed analysis of KASP markers for indica varieties, and variants against the reference sequence were identified, and they were shown in that previous diagram. The mapping rate, genome coverage, and depth were very good. They're equivalent standard, I think, to what's in the Rice 3000 genome project. So we're very pleased with our sequences. We used a bioinformatics pathway to go through a filtering step a set of filtering steps to work out which of those KASP, which of those indels and SNPs were usable as KASP, i.e. did they have good enough quality sequence either side with no repeats and not too many ambiguous bases, and did we have enough read depth to be confident that they would work. So that process generated a large number of potential KASP. This diagram shows how those potential KASP would look when you do pairwise comparisons between the two varieties. So at the top, the right-hand triangle at the top with the numbers in the hundreds shows the number of pairwise comparisons in the original set of KASP before we started. And the triangle on the bottom left with the numbers in the hundreds of thousands shows the number of pairwise comparisons of potential KASP when we'd done that analysis in this feasibility study to look at the sequences across the genomes of these 9 varieties. So we can increase the number of KASP potentially by 100-fold in using this method to identify new KASP. So the next step was then to validate them. So the feasibility study, it identified these KASP across the 9 genomes, and we actually validated it by the end of the feasibility study, which was 2016. We'd validated at least 38 new KASP from that set across the crosses that were being used in Nepal. We'd also used many, many more of the markers that were available in that breeding program to start with, more of those KASP that were available. But the new markers that we identified, there were over 92,000 that are actually in genes that are known to potentially affect gene expression through change in amino acid produced or premature stop codons. So those KASP assays potentially could be really useful if they're affecting traits of interest to breeders. We also increased the rate of identifying KASP from I think it was 1 every 1,000 kilobase pairs originally to 3 every 1 kilobase pairs. And as I said, over 300,000 per pairwise cross on average. We, as I said, demonstrated KASP conversion for any pair of rice varieties. And we used these new KASP to improve the efficiency of the NARC and NARC public sector breeding program and the APL private sector breeding program in Nepal. And we published the findings in this paper in 2018 that's shown at the bottom of this slide. The findings are published along with the code for the filtering of KASP design. So anybody who has access to rice whole genome sequences can use that code to design KASP if they can follow the bioinformatics procedures. But then we went on. We had more funding in 2017 to the present day. That is funding an international project, an industrial research project from Innovate UK. And the idea of this is to enable uptake of KASP by breeders such as yourselves across Asia and beyond to improve rice breeding. And ultimately, this should improve the livelihoods of millions of rice farmers in other rice breeding programs, building on what's already happened in Nepal. The new project had 2 new partners, so NIBG in Pakistan and SKUAST in India. SKUAST is a university and NIBG is a public sector organization, a plant breeding organization. And so they joined the project, and including, we extended the number of target traits that we were looking at. So SKUASP were particularly interested in looking at resistance to Buchanay disease and also brown spot disease. And we also included abiotic stresses for resistance for drought and salinity, and this is particularly of interest to NIBG. So by applying KASP in these breeding programs, we aim to improve the efficiency of selection and adaptation for these stresses, and aim to reduce yield penalties in locally adapted varieties so that we could reduce pesticide use and increase economic benefits in these countries. Just showing you on a map where these organisations are based. So on the far left you have NIBG in Pakistan, and then at the top we have SKUASP in Kashmir in India. Then we have APL in Nepal, Amulbu, and then finally on the right is NARC in Kathmandu in Nepal. Focusing into Nepal, these are the regions where NARC and APL have been carrying out their field screening for BLAST and BLB. Alongside the KASP, after selection, the selected lines have been tested rigorously in the field in both the mid-hills and the terai ecosystems of Nepal, the terai being the yellow areas shown on this map. The aims of the industrial project were to build on the work carried out in the feasibility study to make new KASP assays available for all rice breeders. The pictures on the right show the team at APL collecting leaf samples that they could send to LGC for the full service of KASP genotyping. And the picture at the bottom shows the collection tubes that LGC provide for breeders to send the leaf discs back for DNA extraction. We aimed to generate KASP designs that can be used in a wider variety of rice lines. So rather than just designing them using the 9 genomes that we'd previously looked at, we wanted to broaden that range to make KASP more useful for many, many more rice varieties. We also wanted to identify useful SNPs and Indels that would replace microsatellites. So we focused a lot on trying to design KASP that were near to known microsatellites and other PCR targets of interest, and to run them in assays to actually confirm their use. We also planned to validate 4,000 assays, KASP assays, using genotyping, using lines of rice from the different breeding programs, and we tried to collect rice from a number of extra countries as well as the ones on the project. And we ultimately are creating a database with LGC Biosearch that will allow customers such as yourselves to order these KASP that we've designed directly from them. And those KASP will be available on that database. And in the interim period, I think if you ask LGC, they can help you to choose the markers from our project that will be of use to you. So how did we widen the set of varieties that would be useful that these KASP would be useful for? Well, we used the Rice 3000 or Rice 3K genome project data, which is freely available. The Rice 3000 genome project was run by IRRI, and it has been very successful in gathering together sequences of a whole range of different rice types. So you've got Indica, Japonica, and Basmati, Aus and Boro, and so on, represented in that set. So we chose from that set 119 that represented as many different countries as we could from that set, and a mixture of these different subtypes of rice. And we used bioinformatics to align those sequences against the Indica reference genome. And in the industrial project, we were using the improved reference genome R498 as our reference genome in this project. So we used those along with the sequences of from the feasibility study, our nine sequences, and other available reference sequences that are also coming on stream since the project started. We use bioinformatics to call all the variants possible and to look at where those SNPs and Indels were situated with the correct sequences either side that could generate the KASP designs. Now, as I said before, if you have a target, so the target SNP in this case is shown with the line underneath it, if you compare that between two sequences, so A and B, you may have variation in the flanking sequences that are used in the primer design. Now, if those are a variable, then you can design your sequence to have ambiguous bases. So in this case, the third sequence shown here has the W accounting for the A and T on the left, and the K accounting for the T and the G on the right. But then if you run that on varieties C and D, which have different SNPs that don't match either of those and will not work with those ambiguous bases, then those primers may fail in those varieties, meaning that those primers are not going to be useful in as many varieties as you thought. So you can design better designs to take into account more of those ambiguous bases. And we did that using 134 different rice varieties to try to maximize the amount of ambiguity and to improve the design of the KASP. So we managed to do that using these 119 from the Rice 3K project and 13 additional sequences, 9 of which we sequenced in our feasibility project. And using that, we found 1.2 million assays that were suitable, and the process, the method to do this looked for targets that were available in over 90% of those lines. So they should work in most of those pairwise crosses. And we ensured that they aligned uniquely in both indica and japonica reference genomes. The chromosome map on the right shows the alignment to the indica R498 genome. So all of the potential KASP across all of those 9 original genotyped sequences, the 9 original lines chosen by the Nepalese breeders. So this shows where we've got KASP from this new set that are useful in our crosses for those breeders. So you can see that there are a few gaps here, but as we know, when we look at those sequences, at those individuals, we can fill those gaps if we just use those. But I think this data set, this set of potential KASP assays is really valuable because it will work in more lines than previous KASP designs would have worked in. So it's as a starting point to see if KASP work, it's strongly recommended to try a number of these KASP in the region of interest and see if you can find polymorphism in your parents of interest. And if they work, that's great. If not, then maybe try one of the methods such as the KASP conversion. The other thing we wanted to do was maximize new KASP designs that were associated with target traits. So we used the sequences from the Gramine database for SSRs, for their primers, and other primer pairs that are in the literature that are very relevant to rice breeders. And from these, we again blasted those against the reference genomes to find the sequences and the unique positions in Indica and Japonica. And over 17,000 primer pairs for SSRs were available as potential KASP. There were only 30 that were only found in Indica, and there were 2,000 that were only found in Japonica. So we used that information to generate a set of designs that we would then use for screening a population of rice collected from different parts of the world, which is shown growing in our greenhouse in North Wales here. So the achievements. We have actually validated and shown that they actually work for genotyping 4,867 of the KASP designed using this improved assay design from the industrial project. So we're really pleased about that. We also showed that the success rate for validation increased from 72% of KASP being usable to 89%, nearly 90% in the industrial project. So we're very pleased that these will work, but the caveat is they may not work in every single cross, but they are worth testing to start with. We ended up with genotypes for 3,365 KASP that are useful for rice breeders associated with SSRs or other target sequences. And they were genotyped on the 170 diverse rice varieties that we collected. So that information and the polymorphism between those varieties is going to be made available on the database, which is currently under development by LGC Biosearch. And when it is available, it will be an easy way to check for polymorphism in certain pairs of rice varieties that were the ones we used for the development of the assays, and also to see the alleles that are present in those 170 varieties. But more excitingly, the Nepalese marker-assisted backcrossing program has actually used one of its advanced crosses, and it's got to the point of multi-location advanced yield trials, and one of those is shown on the right here. This is a line that's come from a cross between Sonola Saganda and Loctantra. They were two of the varieties that we sequenced in the feasibility study, and these are aromatic rice varieties that are higher yielding than the parents, the Sonola Saganda parent, and they're very promising, and we're really excited about that and hope they should be released very soon. Pakistan group in NIBG have actually now got data for a genomic selection program that they're undertaking with over 1,000 KASP. So we're hoping that that will lead to a demonstration of using KASP for genomic selection in the future. So NARC in Nepal sent me this slide. They used a KASP marker associated with BLB resistance to improve a cold-tolerant variety called Jumli Marshi that has not been sequenced, and they have demonstrated that they can select using markers associated with known resistance genes that we designed, varieties that will produce grain in areas that are susceptible to, this is blast, BLB, this is blast, susceptible to blast, and they're really pleased that they can improve high altitude rice in this way, which hasn't been done before. In NIBG, they are also very pleased. They've used 215 KASP markers so far for association mapping, and they're working on marker-assisted selection in their Basmati breeding program. They are also, as I said, using genomic selection, and we're hoping to be able to bring more results of that in the future. And the pictures here show their field work and also visits by Dr. Arif to Bangor on the right and LGC in the middle in the UK. In SCUAST, deliberations were held about rice KASP genotyping, and breeders are very enthusiastic. Here are some pictures of breeders in SKUAST taking leaf disc samples for KASP genotyping. And finally, we have visiting dignitaries, including Director General of the Indian Council for Agricultural Research, visiting SKUAST and hearing all about KASP in their breeding programme. So finally, onto the acknowledgements. I'd like to acknowledge the funders, Agritech Catalyst and DFID, Department for International Development, for this project. I really want to thank my colleagues at LGC Biosearch. That's Dominique Darshner, who's here with us in this webinar, and I've not mentioned on the slide, but I'd like to mention Rhianne Gwilliam, who's been really supportive, and we couldn't have basically done this work without the team from LGC, so thank you. Then my colleagues at Bangor, Professor John Whitcomb, who's seen here in the middle on the night shift in Nepal collecting leaf samples, and my colleague Mark, who is our bioinformatician, who has, without his work, we would not have been able to design these useful, wonderful new KASP markers. And then the breeders that we've worked with, there are many more than I can list on here. So Dr. Arif from Pakistan, the team from NARC, so that's Resham and Rambaram Yadav. Then there's the team from APL. I haven't actually put on here the team from SKUAST either, so thank you to the team from SKUAST. And finally to the Agri Epicenter, which is the UK organisation that has helped project manage this project. So now I'd like to hand over again to Marcel, and I'm open for any questions. Thank you.
Thanks, Catherine, for this very interesting presentation, and I would say very important research also towards developing those improved rice varieties for South Asia. I'd now like to do a short Q&A session, and answering questions alongside Catherine will be Darshana Vyas, who is a senior scientist specializing in plant genetics and has enabled of biosearch technology to the global plant breeding community in all species. She was the project manager for Bill and Melinda Gates Foundation, the Generation Challenge program, to ensure the successful application of genotyping as an advanced molecular marker tool for breeding programs in developing countries, which is undertaken for 12 economical important crops. And Dr. is also actively involved in African orphan crops with a successful industrial grant application for rice, conferring resistance. We'll now go over to numerous questions. We have a large amount of questions. Catherine and Darshana, I'll be firing those questions at you. First question.
Thank you.
How to determine there is allelic variation and it's not the same gene in another cultivar?
The allelic variation that I presume we're talking about here is is essential to actually screen the parental varieties to understand the allelic variation, which may not be the same in another cultivar. So that's how you get confirmation that these markers will be relevant in your particular population.
Another question we have received is, do you think that the BH 2.1 is sufficient to screen for aromatic or non-aromatic? The intensity of the aroma matters.
I agree. Aroma is complex. We've shown in at least 2 crosses with our breeding partners that we've designed KASP that work on the same SNPs and Indels that are used in the currently available PCR marker, and they work just as well. The only caveat is that there are some varieties that in some instances So basically, we've got one marker that is what we call the perfect marker, and that works pretty well. But other markers in that region may give a misleading result unless you are absolutely sure that you're using the right parent. So basically, if you may have a parent that gives no polymorphism, whereas you know that it doesn't carry the aroma gene. So as long as you know what your parent's genotype has, then you can use the markers successfully, and the breeders are really pleased with the results of this. Basically, you just need to do a bit of trial and error to start with your parents and the markers that you're choosing before you rely on them for selection. But as soon as you've worked out that they're associated with the trait or in the selected lines, then there's no problem with it. Does that answer the question?
Yes, I hope so. How effective is SSR to define heterozygous individuals in comparison to KASP?
Oh, it's great. You get heterozygotes. Obviously, in early generations, you get a lot of heterozygotes. We basically are using them to show Mendelian inheritance. We basically markers that have weird results, so you don't get the right number of expected heterozygotes. You can usually look at them over several generations and work out what's going on and why. And if they're not reliable, then we can ditch those markers and find other markers. So yeah, you can see heterozygotes very clearly.
Okay, another question is, are all the KASP validated and optimized by LGC?
All of them. Well, no, the answer is no. So basically, LGC do two different types of KASP when you actually start asking them about what you want to do. So they do KASP by design, which basically it will run the design through their Kraken software and say whether they think it will work or not. And then it's up to you to run it. And so you pay for the genotyping. So that isn't fully validated. The other service they have is KASP on demand. So that produces sequences and KASP primers that have actually been tested by LGC and fully validated through their labs. And so if you need to buy assays that you know will work, you have to use the KASP on demand. But in my experience, KASP on design, if you are in the sort of phase of trying to work out which sequences to use, then KASP by design is much more flexible, and you can work out what you really want. And once they're working, then you can go for it. But the beauty of what we've got now is when our sequences become available, when our KASP assays become available on the LGC database, people will be able to just pick and choose the ones that they want to try and test them on the parents. And if they're polymorphic in their parents, then they won't need to go through either of those options, I think. But Dusty might want to clarify on that.
No, that was perfectly explained, Catherine. So it's just the two options for KASP by design or KASP on demand. The KASP on demand is a fully validated assay that we optimize in the laboratories here. That's the main difference.
Great.
Okay, thank you. So if there is no SNP available in the SSR specific fragment, how do you then design your SNPs?
Do you want me to say?
Yeah, go for it, Dusty.
Well, we can both answer it. So the likelihood, especially with the project that we've developed here, where we've seen a massive increase of the available SNPs in the genome, the likelihood of not finding a SNP in proximity to the trait-specific SSR is unlikely. Do you agree, Catherine, or?
Oh yeah, I mean, if you've got very, very closely related lines and you're trying to find a SNP, then you probably won't find one, but that's because they're so closely related. What would be the point of actually using that particular region in a cross. So no, I agree. I mean, there's a huge number of SNPs. It's basically finding SNPs that actually work with the KASP, and as we've shown, there are lots of those that work as KASP.
Mm-hmm. Another question asks, how effective are the KASP markers to identify physical positions in the genome if they are localized on highly repetitive and hypervariable regions?
That's a good question. Do you want to
take it?
Yes, please.
Yeah, I can do it. So again, it's down to ensuring that you've got a good reference genome, which comes into play here. So if you've got a good reference genome, you know the physical positions of the KASP assays, then it should be very effective in understanding the regions where there is high repetitive sequences and hypervariable regions. So for me, KASP can be used like that, but you need to ensure that you have a very good reference genome.
Mm-hmm. So if you're using multiplexing, what is the maximum number of markers that you can use in a well?
So we're talking about looking at multi-allelic SSRs. Is that the question?
Yeah, so the actual question is, what is the maximum number of markers per well in multiplexing?
So at the moment with KASP is a singleplex reaction, but we have developed technologies where you can multiplex SNP genotyping. At the moment it's through our BHQ plex service, where we can multiplex up to 2 pairs of 2 markers in a single reaction. So it's not with KASP at the moment, but we can cover the multiplexing.
Another question asks, the 3.1 markers per 1 kb, is that region specific or is that a genome wide rate?
That's an average genome wide rate.
Mm-hmm. What is or are the blast gene targets that are selected for genotyping?
Oh, there's all the ones that are published. Yeah, I mean, we've got things like XA5, XA13, lots of them. I mean, we've basically tried to target as many as we can find in the literature, and in the obviously in the original cross we had in the original 9 we had target donor parents that carry specific R genes. I don't know if we can go back to that slide. If you don't mind, I'll try and find that slide, which shows some of the R genes that were targeted right at the start of the project. But if they're in the literature, we should have designed KASP near to them. Sorry, it takes a while to go back through the slides. Here we are. Oops. So that shows things like XA5, XA13, XA21, and so on. Does that answer the question?
And I'm sure that if there are some questions that are unanswered, that people can get back to you specifically to the email that was shown on the last slide. Let's try to answer one more question, and then I'm afraid the hour is up. So what would be the procedure to develop an industrial project, and what would be the approximate cost of such a project?
Oh gosh,
that's a big question.
Yeah, so it's international. So it's UK government funding. So you have to have a UK partner, and it depends on the call and what the call guidelines are. You have to have a company partner in the UK, and once you've worked out all of that, it can be. I mean, the sizes range from hundreds of thousands to millions of pounds. Our project in total, I think, was around about £1 million in total, of which some of that is industry contribution and some of that is government funding. So Does that answer the question?
Yes, I think so. I'm gonna try and put the last slide back on the screen, where there is the email address listed. We
Sorry.
still have numerous questions in the chat column available, but I'm afraid that's all the time that we have today. So if you have unanswered questions, please address those to the email address that you see on screen. I'd like to thank very much our speaker, Catherine, for joining us today. And I'd like to thank LGC Biosearch Technologies for making this webinar possible. And of course, a very big thank you to everyone that has been participating and typing questions in the chat box. I hope you found this information very valuable. As I said before, a recording of this webinar will be made available on European-seed.com. Thanks again, and we hope that you have a very nice day.